So tonight we're going to talk about diagnostic tools for worm control. Things like the Camacho System, which I think most of you have probably at least heard of. The FivePoint Check is a new system, and some of you may have heard of it. There's really not a lot of information out on it, but I really like it, and I want to talk about it. And then we're going to talk about what I call the lab tests, how we can look at blood, poop, larva, other things that we look at. Most of those we send away. Some of that we can do at home for ourselves. So that's what we're going to talk about tonight. This is the question that this is the question that we have to ask ourselves all the time. You know, we have our animals. How do we tell if our sheep or goats have parasites? And I think I used the wrong sentence there, but what I want everyone to understand, and I've said this before, they're all infected with parasites. They all are infected with parasites. It's not a question of whether or not they have parasites. They are. And they probably have different kinds. It could be different kinds of stomach worms. We got coccidia. We've got other parasites that can be an issue, or not even be an issue, but be part of what they're carrying at different times of the year of their life. So when we look at a fecal sample, it's normal for it to be positive. You take a fecal sample for your dog to a vet, and if it's positive, he looks positive negative usually, and then deworms them if it's positive. Well, we expect a positive fecal test, whether we're talking worm eggs or the oocytes from coccidia. We expect them to be positive. A negative test is rare. Now, when we're not at a high risk period, like whenever I do any teaching on fecals in January, I hate doing that. Because in Maryland and most of the places you guys are from, the parasites aren't active. They're hibernating. They're in that hypobiotic state. So we do get plenty of negative counts. We did a workshop on the 6th at the Maryland Sheep and Wool Festival. There weren't a lot of eggs. I did one a couple of days earlier in Pennsylvania. We had 2 eggs for like 10 samples. So when it's not a high risk period, we may indeed see negative egg counts, but don't expect to and don't strive to see them. The animals do need parasite infection in order to develop immunity. Remember, the lambs and kids, they're not born with parasites, but they will be exposed as they continue to grow. And the idea is that they're exposed to levels that are low enough, high enough to allow them to develop immunity, but low enough that they don't get clinically parasitized. And that's both coccidia, stomach worms, tapeworms, all of them. There's some parasites they're not going to develop resistance to. The perfect example is that meningeal worm, that deer worm that they're an unnatural host for. Okay, so accept it. All of them have parasites, and it's not bad. It's part of the grand plan. It's part of the grand plan. It's part of their system. Parasites have a role in stimulating immunity. It's just a balance that we're trying to find so that the animals have them because it's normal, but they don't have so many that they get sick. Clinical disease is going to occur at a certain level, and that level is going to vary by species. It's going to vary by animal. It's going to vary by year. When an animal's more highly stressed, it's going to have a problem with parasites, a much less or lower parasite burden. The other thing to keep in mind is not all parasites are pathogenic or even what I call highly pathogenic. I've mentioned before tapeworms. People do find it hard to believe that those nasty, ugly things don't cause a problem. But generally speaking, they're not pathogenic, meaning they're not causing a disease problem. There's a number of the other stomach worms. Whenever we see nematodyrus or esophagostotum eggs in our feces for our goat test, they're there, but they're not really causing problems, both because the level is not very high and they're just less pathogenic than a worm like the Barber pole worm. Barber pole worm is the very most pathogenic worm we have. But at the same time, not all strains of parasites are pathogenic. They've actually, I read this a couple years ago, I don't remember whether it was New Zealand or Australia, but they determined that that not all strains of the Barber pole worm were pathogenic. And we definitely know that's true of coccidia. Is all of the coccidia, there's like 10 different strains that affect sheep and goats. They're specific to either sheep and goats, but not all of them cause disease. Not all of them. So the more important question we have to ask, not whether our animal's infected, because they are, and it's a bad word because infected usually makes us think an animal's got a disease and we need to do something. But we want to know if they're clinically parasitized, they're being affected, they're suffering negative effects, not just death loss, but production loss, things like that. And then if that's the case, do they require some sort of treatment? Do they require implemented treatment? And that's where our diagnostic tools are going to help us answer that question. Because in the old days, we didn't care. In the old days, we said, well, I am just going to deworm them every 3 months. Or I am going to deworm my lambs or kids every 4 weeks during the summer. I'm going to deworm at breeding. I'm going to deworm at lambing. We did not bother to ask if they were clinically parasitized. We might see some throughout the year, probably did, that were clinically parasitized, so we dewormed them again, but in many cases, we just dewormed on a schedule. We still do that, some in the sheep and goat industry. We clearly do it to a large extent in the equine industry. I was just talking to somebody today that boards a horse, and they routinely deworm that horse every 2 months. So that's not doing it because it needs it. It's, again, doing it on a regular routine schedule. That's not a good idea. It gives a false sense of security, and it's what leads us to the resistance issues that we're now dealing with in sheep and goats especially, but also in the other livestock species, with equine not being too far behind. So we've got some tools to help us make that decision. Some of them are things that we do on the farm with the animal, and those are, to me, are the most important ones. We'll talk about those two first. And then how can we do our own fecal testing on the farm to help us, again, with making some of these decisions on managing this risk on sheep and goat farms. And then there's various what I call laboratory, because you send them away. You send them, a vet does them, you send them to a diagnostic lab, a research lab, somebody else does the test, we pay for it. But in most cases, that information is very valuable. And so we can look at blood, we can look at the fecals again, do a lot of different things with the fecals. I have larva down as the third one, but usually that's just still starting with the fecal, is the larva. Okay, so here's the list of all the tests I'm going to talk about tonight. And I've got them separated again by what you can do on the farm. And this is what just about all of us need to do, or at least portions of it. And then if you're having a really difficult time with parasites, you may need to do some of the laboratory testing on the right. Or sometimes just doing one test can clarify things in your program. You notice I got a couple of tests that are in light gray that you can't see as well. I'm going to talk a little bit about a couple of them, but they're really not available. The dipstick test and the Carla saliva test, they're available in Australia or New Zealand. They're not available here. And then you can look at the level or the larva on pasture. I don't really know the specifics of who does that. My understanding that it's quite difficult to do that. But that would be something you could figure out. But simply having fecal egg counts kind of gives you an idea of the infection level on pasture. Mostly the looking at larvae on pasture is done in the research community. You'll know, if you recall, I've presented some graphs that give pasture larvae trends and numbers and that sort of thing. So we're going to go through each of these tests. A couple of them are new, as I've indicated, one new on-farm diagnostic tool and one laboratory test that's relatively new. Okay, we're going to start out with the Camacho system, or people say it differently. Sometimes they put the accent Camacho. And where it gets that funny name from is the gentleman who I guess was the lead in developing it. The system was developed in South Africa for small-scale sheep producers. And it was developed because they were having significant problems with resistant worms. So animals were getting parasitized, the farmers were worming them, and they were dying. So they had a lot of resistance issues. So the researchers developed the Camacho System to look at to assess Barber poleworm infection, which was the primary parasite, and to determine whether that animal needed dewormed or not. And again, it was the name for the man who developed it. Fafa was his nickname, Milan was his last name, and then CHA for chart. So if you ever wonder where the funny name came from, that's where it came from. This slide is kind of busy, but it really shows you exactly how the system was developed. The Barber poleworm, the primary symptom is anemia. They lose blood and protein. They get anemic. We can see signs of anemia by looking at mucous membranes. You can look at the gums, you can look at the vulva, you can look at the skin, but the lower eyelid, the conjunctiva of the lower eyelid, every time I say that, actually I'm pulling my own eyelid down. You can't see that, but it's just, for some reason, I always do that. So you're looking at the color of that lower eyelid. So what they did is they checked a lot of eyes, and they took a blood sample from the sheep at the same time. And so they assigned these clinical categories, 1 through 5. Basically, they're different colors. They aligned them with the packed cell volume, which is the proportion of, and we'll talk about this test a little bit later too, the proportion of red blood cells in the blood. And those are the ranges that you see there, and they assigned the treatment recommendation. So when the animal's not showing signs of anemia, which is in the first 2 clinical categories, and with those packed cell volumes, there's no need to deworm. Unless something else tells you should, like bottle jaw. And then when it has the pale colors and the packed cell volumes 17 and below, those animals are showing signs of anemia, particularly severe in number 5, and require deworming. And then, of course, like everything in life, there's that one in the middle that, should we deworm it, should we not? And the recommendation on that one varies. Originally, the recommendation was to deworm young ones at 3, 4, and 5, and older ones at only at 4 and 5, or goats at 3, 4, and 5, but sheep just at 1 and 2. Because eventually what they did is they developed it for sheep, but then they validated it for goats too. And then the other thing that happened is when the system was brought to the United States and basically came into the University of Georgia and a group of scientists in that part of the country, they validated it all over again, meaning they looked at a lot of eyelids, bled a lot of animals to make sure it worked. So they validated it for sheep in this country, they validated it for goats in this country. And originally the card was fairly large, like you see on the lower right, and now it's a smaller card that's a lot easier to handle, to work with in the field. Now, when you're using the Camacho system, in order to get that card, you have to take a training. This one doesn't qualify because we're not doing anything hands-on. We taught a training at the Maryland Sheep and Wool Festival. I know a lot of folks aren't from Maryland. It doesn't do you any good for me to talk about trainings in Maryland, but we will be having one at Sheepman Supply in Frederick, possibly one in Baltimore County. If you're in another state and would like to get a training so you can get a card, I'd suggest you start with your county extension office or a sheep and goat specialist in your state, or maybe even your state sheep or goat association. Those would be some places to start. Once you take the training, get the card, you want to use the card. You know, you don't just want to look at eyelids, you want to consistently use the card. You're going to need to replace that card periodically because colors do fade. The other thing that's real important is that you use the card, but don't look at it through a tunnel. Still look for other factors that affect whether or not an animal is thriving, whether or not it might need deworming. And that's kind of where the 5-point check eventually leads into. That kind of quantifies that idea of not just using the card as your only decision-making tool, but the 5-point check kind of quantifies it into making it a system. You don't have to retake the class to get a replacement card. One of the things instructors such as myself will do is we will provide the names and addresses to the University of Georgia, which is the sole distributor of the card in the United States. We'll provide that so that you can go directly to the University of Georgia and get a card. If you took the class from me, I'm happy to sell you another one. They're basically just $10. And so the Camacho system is just another tool that you fit into other things that you're doing on your farm. A lot of those things we talked about last week, you know, some of your grazing management, your nutrition, you know, your general management. FivePoint is another tool. It's not like, well, that's all I have to do now. I don't have to do anything except check their eyelids and then deworm them if they need them. No, it fits into an entire program. The benefits of the Camacho system is it reduces the number of anthelmintic treatments, reduces the number of treatments that you give your animals, the frequency of treatment. Because if you see this is a goat, clearly a 1 or 2 score, no need to deworm this animal. In the past, we might have. So you end up spending less money on anthelmintics, hopefully, because while some animals might need deworm more often, the majority won't. The other good thing it does is it identifies susceptible and resilient animals. So whichever way you want to look at it, well, I want to find the susceptible animals and get rid of them, or I want to find the susceptible animals and not save offspring to them, or I want to find the ones that are resilient and make sure I save offspring from them. It's going to identify both. It's not necessarily going to identify the low egg shedders. Chances are if an animal is susceptible, he's also probably shedding a lot of eggs, but it would be possible to have an animal that sheds a lot of eggs but doesn't require treatment because he's resilient. By not deworming all the time and frequently, we're going to increase what they call refugia. Refugia are the worms that are in refuge. They have not been exposed to a drug or a specific drug. And so those susceptible worms are in the population of worms, and they mate with the resistant worms, and we maintain that susceptibility genes within the worm pool. So it prolongs the effectiveness of anthelmintics. So say you wouldn't know this necessarily, but say ivermectin was only 50% effective on your farm. Well, if you started implementing the Camacho system and the concept of selective deworming, you might be able to maintain that 50% for a lot longer. If you continued to deworm frequently and do a lot of the things that contributed to the resistance, then you may well render ivermectin useless in a relatively short period of time. So the whole idea is by deworming less, we are prolonging the effectiveness of the drugs that we have, or delaying that development of resistance. Because one of the things you got to realize is with any drug, anthelmintic, I don't care what it is, resistance is inevitable. Because no treatment, I don't care what it is going to kill everything out there. So if you deworm an animal and it kills 98% of the worms, that's pretty darn good. 2% survived, and that 2% will keep increasing. We've been using the Camacho system at our research center for about 7 years. We do the goat test now, but the year before we did the goat test, we had a study with lambs. We ran 80 lambs on 10 acres. They were out of Katahdin moms and sired by Suffolk, Texel, Dorper, and Katahdin rams. And this chart, the one on the left, shows how many lambs were wormed at each of those dates, and it gives you the average Camacho score. And then the one on the right shows you how often lambs were dewormed. If I compare that to the old system of, well, I'm going to deworm those lambs every 4 weeks, that red line would just be straight across the top. Okay, we only found the need to deworm about 2/3 of them. Oh, it peaked about July 11th. The lambs came in May. They had never been dewormed, and the need for deworming peaked at July 11th. 4 weeks later, we were down to nothing, and we maintained a low level. Granted, every year is different. But what's interesting that is if you look at that graph on the right, about 26% of the lambs required no deworming. 26%. So again, compare that to the idea of deworming lambs every 4 weeks. 26% didn't require deworming. 42% only needed one deworming, and that was most likely at that July 11th. So I've got about 70% of these lambs that only needed dewormed once during the summer. And again, compare that to a system where we might have dewormed them every 4 weeks. If you move to the top, you see just a few lambs needed dewormed quite frequently. There's one that needed dewormed 6 times. You'd know to get rid of that lamb. or you'd know to get rid of those at the top. And this kind of supports the idea, if you remember the 80-20 rule, where it's only about 20% of the flock that's responsible for about 80% of the contamination. So it's a small percentage of the flock that has a lot of problems. And we need to save those animals, like the one that needed to deworm, the ones that need to deworm 4, 5, and 6 times. We save them. We get them through to market, but we don't keep them because they're clearly not resistant or not resilient. In this particular study, what was interesting, it just observation, not scientific fact, but the Suffolk sired lambs needed quite a bit of deworming. The Texel sired lambs didn't, and that was really interesting. Yes, your question about the card. Yeah, they'll maintain that list, and they can see you've taken the training. And I do the same thing if I know you've taken the training. Usually I just believe you if you tell me you had, if you've taken my training. If you haven't taken my training, I won't probably will send you to the University of Georgia. Camacho System, what are the limitations? Nothing's perfect. Everything has, well, it works, but. First thing, it does not save time. You will deworm less, but you need to check them more. Because, again, I'm going to look at that every 4-week system during the summer. I dewormed them every 4 weeks, and after I dewormed them, I didn't worry about them for 4 weeks. Well, if I didn't deworm it, I better check it again in 2 weeks, not 4 weeks. So I'm going to be checking more. We want folks to deworm less, but probably check more. If you're a large farm, got hundreds of animals, that's problematic. It's a lot quicker to deworm everybody than to check them. It's a lot easier. You go to countries like New Zealand and Australia, they don't use the Camacho system on their really large flocks. They look at fecal A counts, not individual counts, not making an individual decision on one animal, but looking at the overall, the flock fecal A count and the pasture contamination. The other thing is, and I think most of you realize this Camacho system is only where barber poleworm is the primary parasite. Now, the good thing is, I think for most of you participating in this webinar, it's the primary parasite. If you don't have a problem year-round, which most of us don't, it's the primary parasite during the summer when parasites are the biggest problem. It does not tell you if you need to deworm for other worms, no. But keep in mind, the barber poleworm is the most common parasite. The barber poleworm is the most pathogenic parasite. It's the one that produces the most eggs. It's the one that's the deadliest. If you recall from one of my earlier slides, I think I showed you the percentage of barber poleworm in our goat test. Always over 80%, very frequently over 90% of the worms are the barber poleworm. The other thing you need to keep in mind about the Camacho system, and really this isn't just true for the Camacho system, it's true for parasite control, period. It's not going to work if you don't know if the drugs work. Because when you truly have a parasitized animal, you need to know that you're giving it something that's effective. And that's a lot of the missing link for a lot of producers. And there are a few tests that you can do to determine whether the anthelmintics work. That's some of those lab tests that I was talking about. That moves us on to the 5-point check and fits Phyllis's question perfectly. There was always that concern that, well, the Camacho system only works in the barber poleworm, and even though that's the deadliest and most numerous worm, what about the other ones? How can I be confident that I'm also controlling the other ones? So what the 5-point check is, and yes, they already copyrighted it in South Africa, to me it just kind of expands the Camacho system to include evaluation criteria for other parasites, pretty much almost all the internal parasites. And it's called the 5-point check because we're checking 5 things. Now, before they came up with the 5-point check, we still recommended that you look at other things. You know, that you never just look at the Camacho system or the Camacho score as the only thing that you look at. And so this, again, it kind of formalizes the idea I'm going to check 5 things to make my decision about deworming. Here's what it looks like kind of graphically. I made this graph. I thought if it's called the 5-point check, it ought to be a star. So I created this graphic, and so the 5 places of and we'll talk about each one of these in detail. Certainly it's the eye for the Camacho score, looking for anemia. The back, here we're talking about body condition scoring. one of the most important things that producers can do, not only to monitor animal health, but also to monitor nutrition. Animal is body condition score. I like to expand that in particular in goats to also talk about the condition of the coat, because I think the condition of the coat, much like a dog or a horse, is indicative of overall health. So I've adapted it and said, and coat condition. And then we go to the back end of the animal, look at the tail, look at the butt, and what we're looking for is evidence of scouring, either current or past. We call it a DAG score. The next place is the jaw. These aren't necessarily have to be in this order, but again, this is just bottle jaw, and we've talked about that before, but the swelling or the edema under the jaw. And the fifth one that is included in South Africa's system is looking at the nose for nasal discharge for nose spots. I don't know if they're considering, I always consider nose spots an external parasite, but technically it is inside the animal in the nasal passages, and perhaps it's a significant problem in South Africa, so they include it. I'm not so much worried about this one, but certainly you still are going to look, that's still something we look at when we evaluate animal health. We're usually more thinking of a respiratory distress, I know, when we look at our test goats than we're thinking nasal spots. Okay, here's a different way to look at the 5 points. But here let's talk about when we look at those 5 points, what parasites are we evaluating? Well, then anemia, again, you know that's the barber pole worm. It can also be hookworm, which is, again, is not very common. It can be liver fluke, and that is probably in some locations that could be common. Body condition scores all of them, because body condition obviously is a very generic symptom of things that can be wrong. It can also just be a result of poor nutrition or production stress. You know, you would expect a ewe or doe that's nursing babies when she weans them that she's not going to be in the best of condition. So you have to relate body condition scores to nutrition to where they are in their production cycle. When we look at the tail or the rear for fecal soiling, scouring, and DAG score, again, that's a somewhat generic symptom in that diarrhea has many, many causes, both infectious and non-infectious. But there are quite a bit of the stomach worms for which that is a primary symptom. Then there we just list them, including coccidia. And my understanding, I've not really noticed it because I've not really had clinical coccidia in a long time in my animals, but that can even cause some anemia. Keep in mind the barber pole worm actually is more likely to constipate them than it is to cause them scouring. But generally speaking, even if they're mostly barber pole worm, they still usually do have a mix of some other parasites. The jaw or the bottle jaw, again, is indicative of barber pole worm, but also liver flukes. In fact, you can Google liver fluke and you can see a picture and you'd swear that's barber pole worm, but it's liver fluke. And then, of course, the nasal discharge would be indicative of the nasal bots. That's a bot fly. It lays eggs in the nasal passages, and the classic symptom is a snotty nose. They may hold their heads down, but ivermectin is labeled for that, but that's something else that you can look at. Okay, so we're going to go through each of the 5 points. Well, we've already talked about the barber pole worm, how it came about, how it's related to the packed cell volume, which is the actual diagnostic test for the barber pole worm. Bottle jaw, these are just some pictures of bottle jaw. Sometimes it's pretty obvious. Sometimes it's a little bit smaller, you know, a little bit harder to notice. If you look at that lamb in the upper left corner, that crossbred lamb, it's not real obvious. It's kind of, you can tell it's there, but you kind of got to look for it a little bit. The picture right directly below it, that you'd have a hard time not noticing that one. The picture of the goat, ironically, is the only bottle jaw we've had in our goat test in 5 years, you know, for hundreds of goats. It's only happened once. So you're not always going to get bottle jaw as an indication to deworm. that's why we have to look at the eyelid. We'll get plenty of ones that don't get bottle jaw that would die if we didn't give an effective dewormer. As far as treating, ivermectin regular treats nasal bots. Liver flukes, I don't remember exactly what's in Ivermectin Plus, but valbazen is actually labeled for the removal of adult liver flukes. Persistent coughs, that's a tough one because that is an extremely generic symptom, just like diarrhea. If you got one animal with a persistent cough, it's certainly not going to hurt to deworm that one. Is it lungworms? Probably not, but you're not going to hurt by deworming, that one animal. Lungworms are very difficult to diagnose except by necropsy. I've had animals that have coughed for weeks and weeks and then they just stop. So I just stop listening. Can bottle jaw also come from lack of mineral? There can be other causes of anemia, and obviously it can be an iron thing. I don't know that it's from lack of mineral, but I guess I can't say for certain that some sort of mineral deficiency couldn't factor into that. Lab results that show coccidia or strongylus, they're just saying coccidia, they don't know what kind. And then the strongylus, they're just saying the stomach worms, they don't know which kind. Because when you do a fecal egg count, or when you look at fecal eggs, you cannot tell. And we're going to talk about fecal eggs and some of the limitations of that type of testing. Because nothing's perfect. It's just kind of when we're able to combine a lot of these different things. It's like being a lawyer. Your job is to come up with the preponderance of evidence. And so I think when you look at some of this diagnostic tools, you're trying to come up with a preponderance of evidence to help you decide whether or not to deworm that animal or whether or not to, to change some sort of management plan. Body condition scoring, again, I think this is just one of the most important things that we can do. There's plenty of fact sheets out there on body condition scoring. The thing I always tell people is to be consistent. Same person should do it. Realize that we have differences on maybe what we think is average or what we want to be average in our herd or what kind of condition we want them to be in. Realize that there's going to be a different feel between a goat and a sheep, between a dairy goat and a meat goat, between a hair sheep and a wool sheep. Keep in mind that both goats and hair sheep to a secondary extent fatten from the inside out, so we don't want as much body condition on those, whereas a wool sheep or most other meat animals, they do fatten on the outside. When a goat or hair sheep has a lot of body condition on the outside, they're pretty darn fat on the inside. No recommendations here in terms of whether or not you should deworm. This is just more information to use to factor into that decision. So basically the information that you'll read about body condition scoring is how to assign a score. And we do use half scores as well. And we want our sheep, really when you think about it, to be between 2 and 4. Between 2 and 4. We want to stay away from 1 and 5. When we breed, we want them to be 3, 3 plus. An animal that's a body condition score of a 3, 4 is going to tolerate parasites much better than one that's a 2. And obviously one that's suffering parasites, if it's kind of more of a subclinical, it's going to lose weight. As far as how soon do you expect an improvement if you treat an animal with an effective anthelmintic, and keep that in mind, effective. And that effectiveness could vary. It could be 50% effective, it could be 70, it could be 90. Some are going to improve pretty quickly, some are not. Very often it takes us, just at our goat test, sometimes in 2 weeks they change one score. Last year when we were using levamisole, we actually would see 2 score improvements in 2 weeks. I've done lambs before. I only spot treat lambs on occasion, and I've seen remarkable responses practically overnight. For those that aren't responding and aren't really doing well despite your treatment, supportive therapy is often a good idea, and red cell is something that is good to give cautiously with sheep because it has copper in it. It's a horse nutritional product for horses. We do give it to our goats in the goat test when one doesn't seem to be bouncing back from the treatment very effectively, or one that's a 5 that's really, a 5 Camacho and maybe also some other things. red cell, again, it's a product, a nutritional supplement for equine. Comes in like a gallon jug. It's just a great thing is supportive therapy. I'd probably rather use something like that than Nutri-Drench. These are just some pictures of some nasal discharge. The upper right shows you kind of the cycle of that parasite. Picture below shows you what the nasal cavity looks like. Picture on the left is just the only picture I had of a snotty-nosed goat. In no way was that nasal bots probably, and I don't even think this goat had any nutrition, had any type of distress at all. I just thought it was a cool picture, and we needed a picture of snot. So those were the 5 things for the 5-point check. Camacho score, bottle jaw, the ones we've always talked about. But now, pulling in body condition score more, coat condition maybe on the goats. Sometimes the condition of wool is also indicative of the health of an animal. A real open fleece. So we really can look at the coat of sheep as well. Now let's move ourselves into what we call the laboratory tests. Things that chances are you may collect the sample, but you're probably going to send it somewhere. I believe most vets would be able to do packed cell volume. And what packed cell volume is, and they call that PCV, they call it blood hematocrit. It's the portion of the blood that's red blood cells. So it's a measure of anemia, which again is the primary symptom of Barber poleworm, could also be the symptom of liver flukes. It's a measure of resilience. Animal could have tons of eggs in him, but if he still has a high packed cell volume, he's tolerating that infection. If you read much research on parasites where they're testing different anthelmintics or natural treatments, the researchers will always take blood samples and determine the packed cell volume. The higher the number, the better. Sheep tend to be, on average, higher than goats. Again, the table shows the Camacho, how Camacho was developed. It's the same table. And realistically, the diagnostic test for the Barber poleworm is not a fecal test, it's a blood test. And a lot of research that you read will actually use 15, and this is percent, as a cutoff point to deworm. A cutoff point to deworm. That actually fits somewhere between 3 and 4. But the packed cell volume can actually be an indication of when I need to deworm, when I need to deworm. And so you could have that done at a vet's office. But guess what? The Camacho system gave us an easier system to estimate that. Is it perfect? Is absolutely every animal going to fit into that table? No, you're going to have some outliers. But the whole purpose of validating the system was to figure out, well, how many false positives and how many false negatives did we get? And for the most part, the system does work. Yes, there will be outliers. But for the most part, the Camacho system is an indicator, a very pretty accurate indicator of packed cell volume. And the way we get around the idea, well, are there some outliers? Again, we don't just look at Camacho score. We look for bottle jaw. We evaluate body condition. We evaluate DAG score. So that can help us find some of those outliers. Fecal testing is what most people are familiar with or have heard of, and a lot of people really want to do it. Well, there's different uses of fecals. First, we can identify worm eggs. We can determine if there is an infection. You know, again, it's normal to have an infection, although certain times of the year there may not be any eggs. More importantly, the level of infection, because if it's normal to have infection, it's normal to have worms, normal for them to lay eggs, it's the level that's important. There's a couple of ways to look at fecals in that respect. There was an earlier question about results not telling exactly what type of parasite they are, the specific ones. And there are some tests that we can use to determine what type of parasites we have. Yeah, we can tell stomach worms in general, we can tell coccidia, we can tell tapeworms, but how do we tell the specific stomach worm? And then probably, to me, the most important use of fecals, and again, the missing bit of information for most producers is, does the anthelmintic work? Is it still effective on your farm? If I have a sick animal and I deworm it with X drug, is it going to kill the worms. Fecal testing, just your basic, let's look at the fecals on a slide. Two systems, what I call a qualitative and a quantitative. A qualitative is what most vets do, what many diagnostic labs do. I call it a simple fecal flotation, no different than you might do for a dog. Different ways to do it, certainly. I mean, if you do it, more than likely you're going to use just a regular slide with a cover slip. You're going to know general kinds of eggs. Again, the stomach worms, the strongyle, those are the round worms. The coccidia, they're smaller, they look different. Tapeworms are kind of triangular shape. So if you can tell the basic worm type eggs, not the specifics. Again, you can determine if there is any, the existence of an infection, but really only the general level of infection. Obviously, you can tell the difference between a couple of eggs and lots, but it's not really any more specific than that. A few people or a few places will use systems like how many eggs they see per field of view, and I think that's what our diagnostic lab does. They don't count them, but they count. They kind of rank kind of an infection. Their most common one is too numerous to count. I get a kick out of that because too numerous to count could vary for all of us. I'd probably get tired of counting after 5. The other is what's called an eggs plus system. You can actually, Bill Shula from Ohio State describes that in one of the newsletters. That's kind of an interesting system that it attempts to quantify it without actually counting them. The quantitative, we're actually determining an egg count. Eggs per gram of feces. This requires weighing the feces and measuring out the flotation. We can still differentiate general egg types. We can't get any more specific, but we're determining the level of infection. And there's what the McMaster slide looks like. And I will answer that question about where you can get it. And I just want to basically highlight that this is where you need to be. You need the quantitative one. Because if you take a fecal sample to a vet or to a diagnostic lab, it's not a question of whether they have worms. They tell you, oh, I found worm eggs. You go, duh. More than likely, again, if you're in the risk period, you're going to find them. So you need to know how many. So buy your vet a McMaster slide for Christmas. Ask the diagnostic lab. We need to do quantitative fecal egg counting. So what do you need if you want to do them on your own? Because fecal egg counts, they're both a farm test and a lab test. You can send them off and let somebody else do them. We do that for our goat tests. I don't do fecals. I send them away. But you can do them yourself. And what you need to do them, here's just the basic things you need to do them. We've taught classes on them. Langston University, where Steve is from, has taught classes on them. You can see kind of some tutorials on the web. You need a microscope. Basically, you need one that's 100x power, which the eyepiece is usually 10. Most people get microscopes, like a student microscope, with about that'll go up to 40, so you'll be up to 400, but you really only need about 100. It's very useful to have a mechanical stage, which is really just a little arm that helps move the slide around so that you can easier count them. You need a flotation solution. You can make it yourself. In fact, the homemade ones I think are better. You can also purchase them. Out of convenience, I tend to purchase them, so it's not one more thing I have to do before the workshop. I usually purchase them from a company called livestockconcepts.com. Somebody had asked a question about where you get the McMaster egg counting slide. It's a Chalix Corporation, which kind of specializes in parasitology on equine. They're one of actually 2 sources. I believe there's a second source of those slides now. But if you just type in vetslides.com, you will go to that website. They actually sell a kit with a couple of slides, and they actually have a technique where, or they recommend a technique where you don't actually weigh the feces, but you determine the weight by volume displacement. Then you need some miscellaneous, inexpensive supplies. You need a scale. You know, I buy scales off the internet for a couple of dollars. They're just a little digital scale. You need something to measure out the flotation solution, the volume of it, like a beaker. You need pipettes or syringes to transfer the solution, the fecal slurry that you're going to make, and put it into the slide chambers. You need vials to mix the solution, craft sticks, popsicle sticks to smush the poop, something to strain. Part of preparing the sample is you will strain it. Now, this is one technique. There are different techniques of doing it, but cheesecloth or a tea strainer or something like that. That's what you can, so you can do that. what does the information tell you? You do a fecal egg count on an individual animal, and somebody sort of asks that question. If you do one animal, well, if you do one animal, there's the fecal sample from X28. That was one of the goats on test. It tells you what the potential burden is in that animal. It may or may not be correlated with highly correlated with the number of worms or the severity of disease. It's a good indication of what they're dumping on the pasture. You know, if you've got a lot of high egg counts, either because you do a group one or you do a bunch of them and they're high, well, you can be fairly confident that they're dumping a lot of eggs out into that pasture, and then those eggs are potentially hatching and developing into infected third stage larvae, which then gets ingested by the susceptible animals. Probably the best use is to determine the level, whether the drug works. And it's kind of the same thing. One animal is not going to tell you a lot. Not going to tell you a lot. What we primarily use them for in our goat test is to look at genetic differences in the bucks on test. I'll pull 9 samples throughout the summer. And so if I've got a buck that has low egg count 9 times, I feel pretty confident that that's a pretty resistant buck. The question about one sample versus 3 or 4 of the same animal, you're increasing the accuracy the more samples you do from the same animal. Just like the McMaster slide itself, by having 2 chambers, that increases the accuracy. If you had 3 chambers, that would increase them. We often ask that a lot and when we get the data back, but it becomes an issue of what's practical. I can't send, if I send 3 or 4 from the same animal, that's 3 or 4 things I have to pay for. So I guess I look at accuracy on ours is I've got 9 samples from the same buck, and if he's low all summer, that's probably pretty indicative that he's resistant. But the more you do, yes, the more you do, the more accurate it's going to be. Because definitely one of the limitations is the technique itself. Fecal egg counts aren't definitive. Personal opinion is, if I've got one goat like I do in this picture, I wouldn't do an egg count to determine whether he needs dewormed. When I think of egg counts, I think of doing multiple animals and multiple times, because they're not always well correlated with disease. We've got a lot of data that shows we can have a high egg count and the animal not be treated, and vice versa. And just because it has eggs doesn't mean it needs treated, nor does just because it doesn't have any eggs or oocytes mean it doesn't need treated. Think of the L4s, which are basically immature worms. They suck blood but don't lay any eggs. So you can have a low egg count, but you could have plenty of those in there sucking blood. Coccidia, you can get disease before you actually get a lot of oocytes. So you want to be careful that you don't let fecal egg counts give you a false sense of security either way. I almost look at them as part of the 5-point check. If I checked those 5 things and I also had a fecal egg count, that would be more information, more evidence that would help me make that decision. If you're getting a McMaster slide, I think somebody mentioned, go ahead and get the green slides. They're a lot easier. They're a lot easier. Oops, I skipped the slide. There's a lot of words on this slide, and you can read them at your leisure, but there are a lot of limitations to fecal egg counts. A lot. And just, it doesn't mean they don't have value, because they do. You just got to consider some different things. Still, we're not being able to tell specifically what type of stomach worm we have. We're still not able to tell that. We have differences in the parasites themselves. Barber pole worm lays a ton of eggs. Other ones don't. So keep in mind there's just a lot of different variables, with human error being the last one. I always joke that our fecal egg counts are done by graduate students, and how possibly accurate could they be? I was a graduate student. And the fecal consistency changes the egg count. It's not an exact science, but it's like a lot of things in life. Sometimes it's the best we can do, and as long as we keep it we don't think of them as definitive, they can be really useful. We're going to talk about the fecal egg count reduction test, which is the question we have up there right now. This, I'm a little bit reluctant to show this slide, but I still like to. I found this out of a publication in Australia. It's hard to find this information because most good parasitologists will not tell you a trigger, a certain egg count at which to deworm. And I still want you to understand that it's not as simple as this. For example, this shows that for the barber pole worm, 2,000 eggs per gram is clinically significant. Keep in mind you could have parasitized animals below that. And that, again, looking at the bottom, it says consider the nutrition and physiological status and age of the animal. All those things have to come into play. An older animal can tolerate more than a younger animal. There's so many different variables, and that's why, again, the fecal egg counts are useful, but we gotta use them in the proper way. But you can just look how much, looking at the sheep parasites. Again, the barber pole worm is a very prolific egg layer, and then you get down to the Nabonidus, you can have problems at very low egg counts, and so you always got to keep those kinds of things in mind. The fecal egg count reduction test, what Gail is asking. This is the first and most widely used method to determine if the anthelmintic works. And there you kind of see a close-up of the McMaster slide. You can see it has a green grid on it. You fill the 2 chambers. The lines help you count. Theoretically, you could do it without this slide, but it's a lot easier to use this slide. When you want to check for the efficacy of a dewormer, you do before and after fecal egg counts. You collect a sample, deworm, and then 7 to 14 days past treatment, you check again. And that 7 to 10 days, there can be a little bit of variation depending on which dewormer you're checking. And Steve Hart, who's on our chat box, Steve's done a lot of this type of work. And if you have for a specific dewormer, you would want to check. Again, if I did one animal, I don't know how useful that would be, but like when we do our 70 goats in the goat test, I'll have quite a variation if I look at individual animals, but when I look at the whole group as a whole, I can kind of figure out how effective the treatment was. We do this every year in the goat test in that we deworm them when they come in order to get them on an even playing field, and then we deworm them 2 weeks later. I'm sorry, we take a fecal sample 2 weeks later, so I know how effective the treatment was. Last year it was only 35% effective. Previous years it was 95%. Ideally, you have controls where you test ones that you treat and you test ones that you don't treat. And that way you can attribute the differences to the anthelmintic treatment, not to something else. In an ideal world, you have that. And ideally, you do at least 10 animals. EPG, yes, is eggs per gram. I've not heard 21 days, but I've always heard it 7 to 14, but I'm not going to say that can't be valid. Is that a Vermont? If that's a Virginia Tech and Ann Zajac, it's probably correct. All right. Fecals actually can be pretty laborious. That's why I send them to Delaware State University instead of doing them ourselves. You have to do them for every drug. You have to do a bunch of animals. The alternative to this is a test called, well, in the industry it's called the drench test. It's copyrighted. It's basically what they call a larval development assay, or an LDA. It's an in vitro, in petri dish test for the detection of anthelmintic resistance. It uses the larva. And it tests for resistance to all the anthelmintics from a single sample. And this is just a couple of guidelines. They'll tell you how they want 10 animals at least. They want, ideally, they want at least 500 eggs per gram. You can't really test it if you don't have enough eggs. And of course, to try to hedge your bet, you will try to, even though I'm saying they're not perfectly correlated, you're still more likely to find a high egg count in an anemic animal or one that's scouring. This test, to my knowledge, is only currently available through the University of Georgia, the same lab that has the distribution rights on the Camacho card. It's an expensive test, but you know what? 100-pound market lamb's worth $220 right now. So just losing one lamb or one goat is pretty costly, and And you'll get back a report that'll tell you basically the percentage of efficacy on the different dewormers. Okay. An earlier question asked about, the lab results said strongyle eggs and coccidia. And that's because the strongyle eggs all look the same, the coccidia eggs all look the same. You have to hatch out the larva. And then even then I think it's not still wonderfully easy. It's tedious. But they are identified from their larva. What we will do in our goat test is we will collect the in addition to the individual sample, we'll collect a pooled sample from every third goat. We'll collect it randomly. We'll send that sample off. It gets prepared a little bit differently, and they will hatch the eggs and tell us what percent barber pole worm, what percent trichostrongyles, what percent nematodirus. They'll tell us the percent, and that's why I say the data on our test always comes back very high barber pole worm. It may start a little more mixed than that, but it then turns all barber pole worm. And I'm trying to think whether we pay about $50 for this test. If you were concerned that barber pole worm wasn't the major problem, maybe you had something else in there, this would be a test that would help answer that question. Here's just a graph from our test. This was from 2009. Last year it was so dry we didn't get a good measure of parasites, but you can see we were always above 80% on barber pole worm. And by the time we hit the end of the summer, which in 2009 was when we had the highest worm problems, was all the way at the end of the test, we were darn near 100% barber pole worm. And so that test really provides us with some valuable information and not really that expensive. A new test came out, and I believe it was last year. It's called the lectin staining test. I don't think that many people are using it. You send a fecal sample. I can't remember exactly what the cost was, but it tells you the percentage or proportion of eggs that are barber pole worm. And really, you don't need to know the other stomach worms. You just kind of need to know the separation between the ones that cause scours and the barber pole worm. And it uses a peanut. It involves peanuts or whatever. You only need a small amount of feces. And the University of Georgia does this test, but also Oregon State University. And that's, I think, where the picture's from. So that's a new test that's available that would tell you the portion of barber pole worm. And what might be interesting is if you felt you could justify the cost, is to do that test at different times of the year and see what that portion is, or to do the other one, where they culture them. Winding down here, I always like to talk about what we can't have. There's a couple of recent tests that have come out within the last few years. I think the one on the left last year that our friends in Australia and New Zealand will develop because their sheep industries are so much larger, so they can afford to commercialize these tests. The first one that came out was the one on the right, which is a dipstick test. And it's for the barber pole worm. You prepare the fecal sample, and then you put the stick in it, kind of like a hemoccult test or a pregnancy test, I don't know, and you can determine the level of infection, and I presume it makes a treatment recommendation. Test isn't available in the U.S. I don't know if there's any efforts whatsoever to get it in the U.S. The saliva test came out last year, and it measures antibodies in the saliva. I did have conversation with this company, and they were in conversation with an American company to try to commercialize this test. This would also be a useful genetic tool to be able to select the animals that are resistant and more resistant, more resilient. Because I think a lot of people recognize that one of the ways we're going to solve the parasite problem, because it's not like drugs are forever going to be there or always be new ones, is that we need animals that can tolerate better. And genetics, of course, takes a long-term commitment, but our buck test is specifically trying to identify bucks that are resistant and or resilient to parasites. And the bucks, our best bucks, are both resistant and resilient because they have to be for us to consider them the top in the test. And they're a good source if you're having parasite problems. They're a good source of bucks that will have that resistance. even within your own farm. You know, again, using the Camacho System, using fecal egg counts, you can find those animals in your own herd, and we need to move those ones out that need treated. We certainly don't need to be saving their offspring. We certainly don't need a male. You know, you may be a little bit more forgiving on a female that needs treated. I find it interesting. Actually, people will tell me about how they cull a female because she needs treated. While I don't disagree with culling a female that needs treated a lot, it's really the male that you got to be hard on, because he's spreading his genetics over far more animals, and your male needs not to need deworm, needs not to be shedding eggs. That's where the greater emphasis needs to be, not on how big he is, not what he looks like. But if you're serious about keeping your animals alive, keeping your costs down, trying to make a little bit of money, you need males that are resistant and resilient to parasites. I'm not saying that it's not important in the females, but you can tolerate more on the female. It's that male that needs to have these traits. And there's some programs, you know, like our buck test, that we're trying to get at that. They're trying to develop EPDs on parasite resistance in Katahdin sheep. In countries like New Zealand and Australia, you can purchase animals with EPDs on resistance. You could actually run your own little buck or ram test on your own farm, and you could pull fecal samples from them, and you could create some kind of index. You could do a lot of this stuff on your own. You could improve your animals. And again, the emphasis needs to be on that male especially. I'm not saying the females aren't important, but the male. The male, he's going to spread his genetics over a lot more animals. And it doesn't matter how fast his kids grow if a lot of them die. And when they've looked at males in research, they find tremendous differences, sire differences, when they look at parasites. Tremendous sire differences. And I think that concludes this evening. Our last webinar will be next Thursday, and we will focus on treatments. I write anthelmintics, but we're going to talk about, basically focus on the idea of, well, I did all these things, but I do need to deworm a few. I need to salvage treat some to save their life. I need to, some of them are clinically parasitized and really suffering production, and I need to take care of those. And so what do I use? You know, how do I use the anthelmintics that are out there? What other things might possibly help with treatment? What things might be not available now but perhaps become commercially available?